Welcome to this webinar, Advancing High-Throughput Cardiac Screening with Physiologically Relevant 3D iPSC-Based Models. I'm Diana Spencer, Senior Digital Content Editor at DDW. Balancing assay throughput with physiological relevance is essential for improving disease modelling and compound screening in preclinical drug development. Induced pluripotent stem cell technology has revolutionized early drug discovery by integrating human biology, enhancing predictions of drug safety and efficacy. However, achieving both scalability and consistency in high-throughput applications remains challenging. This webinar will introduce Ncardia's solutions, which streamline high-throughput screening while maintaining physiological relevance. At the core of this is Ncyte Heart in a Box, a physiologically relevant 3D cardiac microtissue seamlessly integrated into automated high-throughput workflows. In addition to advancing cardiotoxicity studies, the model can be implemented in cardiac disease research. Speakers Mariana Argenziano, Associate Director Manufacturing Technology at Ncardia, and Richard Davis, Assistant Professor at the Leiden University Medical Center, will describe the development of a 3D cardiac microtissue model of catecholaminergic polymorphic ventricular tachycardia. This case study demonstrates how the platform can be leveraged to advance cardiac drug screening, providing insights into its translational potential and impact on drug discovery. Dr. Argenziano earned her Ph.D. in molecular biology from the University of San Martin in Buenos Aires and has over a decade of experience in stem cell biology, having conducted research at several leading universities in the US. Her expertise spans iPSC differentiation into cardiac, neuronal, and other cell types, as well as disease modelling and biobank development. Richard earned his Ph.D. from Monash University and has received prestigious awards throughout his career, including an ERC starting grant and Dutch Video Award. Next to his leadership at LUMC, he is a member of the Leiden Renew Node for the Novo Nordisk Foundation Centre for Stem Cell Medicine. His research focuses on modelling cardiovascular diseases using human pluripotent stem cells with models ranging from 2D cardiomyocyte cultures to complex 3D systems integrating vascular, stromal and immune cells. I will now hand over to the speakers.
Thank you for the introduction. My name is Mariana Argenziano and I will present this webinar related to the advances using cardiac microtissues. So to introduce Ncardia as the provider of this webinar, we are having headquarters in the Netherlands, in Leiden, and we are an iPSC provider, and not only for cell supply and custom iPSC services, but at Ncardia we also take care of regenerative medicine process, such as process development and cell production, and also discovery services. Ncardia has been an iPSC provider for more than 15 years, and one of the main characteristics that shows the differentiation of Ncardia is the use of bioreactors for cell production, when we can show high robustness and high reproducibility of our projects. But also we have been working with many different clients in biopharma, as well as we have a proprietary CRISPR technology. And we are going to present some of this technology today related to the cardiac microtissues. But of course, if you have any other questions about the other services, please feel free to reach out to us. So going back to the cell supply, I will go over some of the products that at the moment that are available at Ncardia and that we are going to describe throughout this webinar. One of the products and the most prominent and the one that is going to be presented is the cardiomyocytes. We also offer ventricular and atrial cardiomyocytes. We will also discuss some of our Ncyte endothelial cells. And we also can offer microglia, astrocytes, smooth muscle cells, and some others that might not be listed here. And the star of the show today is going to be the Ncyte Heart in a Box. Of course, the products are not always out of the shelf like these ones. There can also be some customization. In many cases, we can differentiate different cell types using different protocols, customize the vial size, use donor material. Many times using patient lines is pretty much of interest when testing different disease modeling or drugs that are targeting a specific disease. And this can be also done using not only reprogrammed material from patients, but also gene editing using CRISPR. So going back to the cardiomyocytes that I already mentioned, I will describe how we make these cells, what is the potential of these cells, to then move towards this webinar. So the Ncyte cardiomyocytes are I would say the oldest product that we have at Ncardia. As the name might say, Ncardia has started with cardiomyocytes, and then we expanded to many other cells. So these cells have been in the market for a long time. We have made already quite some batches, and we know that this product is very robust. So we get in these cardiomyocytes over 90% troponin T. I'm showing in this flow cytometry 98%, and most of our batches are around 98-99% positive for troponin T and MLC2B. That is a marker of specific for ventricular cardiomyocytes, but of course we look at other markers also when working with atrial cardiomyocytes. These batches are very robust. Within the different batches, we see the same numbers. We have already differentiated more than 10 batches of these cardiomyocytes, and the numbers are always very close together. We also see a lot of reproducibility in terms of the multi-electrode array data that we obtain in terms of intra-well variability, inter-well variability. Looking at the picture on the left, we can also see that these cardiomyocytes present a very typical morphology for cardiomyocytes, showing expression of alpha-actinin and troponin T with very nicely tubules. And of course, they respond also to drugs. There are drugs that are very used in the cardiomyocyte characterization, like nifedipine, isoproterenol, or dofetilide. In this case, nifedipine, that is an L-type calcium channel blocker. As we increase the dose of this drug, we can see how the field potential duration decreases, not only on the side of the screen where we can see the traces, but also on the bar graph. As this field potential duration decreases, at some point, of course, the cells also go into arrhythmia and cardiac arrest. So this is very common in cardiomyocytes, in adult cardiomyocytes as well. As I mentioned also briefly during the introduction, we differentiate ourselves in bioreactors. Bioreactors offer a great advantage for many different reasons, not only the fact that we can obtain a lot of cells. In this case, we're talking about 10 billion cells per batch in only one bioreactor. This process takes about 6 to 8 weeks, so it's very fast and you can obtain a lot of cells, but also it helps us to really control our process because we can control absolutely every parameter of the differentiation, from the oxygen, the pH, the temperature, and all the metabolites going into this bioreactor. So it gives us a lot of control on the process, making the process very reproducible. So we show a lot of batch-to-batch consistency and purity and functionality as well. And these volumes, of course, not only increase the efficiency, but also decrease the cost. So comparing with what we see in other suppliers, about 200 million cells per batch, comparing to the 10 billion cells per batch, we see that when people are running high-throughput screenings for the drug pipelines, then cardiomyocytes can cover more than 2,000 plates and 4 million compounds with only one batch of cardiomyocytes, eliminating the need for revalidating, and of course, speeding up the process and increasing consistency. So, of course, given that we had these cardiomyocytes, and these cardiomyocytes have been really used in the market for cardiotox screening, for safety screening, we also saw the need of improving the systems, mostly with the recent FDA announcement that the 3D microtissues and the 3D iPSC-derived tissues will be used for screening. The Ncyte Heart in a box is a new model that includes several cell types in order to reproduce more physiologically the characteristics of the heart or the electrophysiology of the heart. So we know that there are certain challenges and opportunities, of course, in every system that we use, one of them being the 3D cardiac models. So we know that these models bring the possibility to study many different things, the cell-to-cell interaction. They also help maturation of the cardiomyocytes. We know that cardiomyocytes coming from iPSCs sometimes can be a little bit immature. And they also offer a more complex in vitro environment that can reproduce more physiologically the functioning of a heart. But there are also some challenges. There are the high variability of these models, of course, because we have to seed many different cell types and then in different ratios. There's limited high throughput if we need to, limited throughput if we need to do this by hand. And of course, there's variability and high complexity for the end user. So at Ncardia, we have thought about these things and came up with the call Ncyte Heart in a Box. This heart in a box is a 3D cardiac microtissue designed specifically for high-throughput screening. So it's made of 3 different cell types that I have presented a little bit, and we're going to go through the other cell types as well. The Ncyte ventricular cardiomyocytes. It could eventually also use atrial or another type of cardiomyocytes, endothelial cells, and cardiac fibroblasts. And it's specifically designed to be used in a 384 well plate format. So this helps really use it in automated platforms and bring, of course, a high scalability of this microtissue. So as I described the cardiomyocytes, I also will go quickly through the other cell types just to explain how we have characterized these cells and how do we know that we're working with the right cells that we know for this kind of model. So we work with the Ncyte endothelial cells. These cells, unlike the cardiomyocytes that have been in the market for a long time, were released last year as one of the Ncardia new products. And they also show high purity with over 95% of marker expression like CD144 and CD31. We also look at functionality of these cells. Of course, function is equally or more important than identity. We look at the uptake of ACLDL, that in these cells is quite positive, as you see by the red dots. And we also look at different assays, for example, barrier, epithelium barrier, and also sprouting assays. So in figure C, what we can see is that when these cells are forming a tight barrier, we see that the resistance of the cells is quite high because, of course, the current can go through. But once we treat the cells with toxic compounds that might somehow disrupt this barrier, the resistance, of course, disappears because there's holes in this barrier, and then there's no continuity in between the cells. So this is an easy way of seeing how the cells are dying or how the cells are being affected by different compounds in a cardiotox assay. In the lower graph, in graph D, what we see is the sprouting assays. This has been done in microfluidic devices, and we are comparing to the Hubeck cells that are the industry standard for endothelial cells. So what we see is that these cells, of course, in the control, they can sprout, they can form vessel-like structures, and as we treat with drugs that would inhibit the development of these vessels or these structures, we see that in a dose-dependent manner also this sprouting or these vessels are affected by it. In the case of cardiac fibroblasts, these are very simple cells, but they play a really important role in the human body, mostly in the heart, right? The cardiac fibroblasts are generating a lot of extracellular matrix and providing support to the cardiomyocytes. And the specific characteristic that the cardiac fibroblasts have, unlike other fibroblasts, is that they also come from the endoderm, from the mesoderm, from the same progenitors of the cardiac cells. So they are expressing all the common markers of fibroblasts like vimentin or collagen, but they express certain specific markers that are important for the heart, like connexin 43. And connexin 43 is very important, as you see in green by the little dots. These are proteins that will form gap junctions, and these gap junctions will also connect the fibroblasts to the cardiomyocytes, allowing the syncytium and a nice beating and connection with the interaction between the cells. So how do we make these microtissues? It's quite easy, I would say. It's relatively easy. So the cells will come in a vial, and the cells can be thawed and plated for a few days to allow for recovery. This will also allow the researcher to align with their experiments. They can culture the cells for 3 days or for a few more days if needed, depending on the needs, on the schedules. And at the day of harvest, the 3 cell types will be harvested and mixed in a certain ratio. The ratio that we recommend in our application note for the specific Ncyte cells is a 70% of cardiomyocytes and a 15% of endothelial cells and 15% of fibroblasts. But of course, this could be adapted depending on different cell types. And a solution is prepared, a suspension is prepared and seeded. The cells in general are beaten within a few days, but we recommend that they are used after day 14, when all of the cell connections will be formed. Of course, the endothelial cells will arrange in a vessel-like manner, and all the structures inside the microtissue will be, of course, resembling more that one of an adult cardiac heart or cardiac tissue. These tissues can also be used until day 21, so they also allow for experiments that are quite long or can take a few days. So showing here how this automation actually of the microtissues helps, of course, with making this a much more efficient process. Showing that when we do this automation, we decrease not only the time that it takes to form the microtissues, to refresh them from hours to minutes, but we also increase the reliability of the process, showing that we have less than 10% intra-well variability in these plates, and also the interwell and interwell variability for the volumetric assays is less than 5%. When we look at the compound response, they behave pretty similar to those that are manual, but of course the reproducibility is much higher. And it's always nice to see a robot working. So looking at the identity and morphology of these microtissues, the cells look, the microtissues show all the expression of these tissues. If you were to look a little bit deeper, you can also see the structure of the sarcomeres, for example, but we can see that the cells are arranging in a very heterogeneous way in the microtissue by looking at picture A, and it's not that they're all in certain areas or oddly distributed. They distribute evenly. And in general, we use less than 10,000 cells for microtissue. So they do require a quite low number of cells. Seeing that the microtissues can also beat very nicely after a few days in culture. This is already a picture of a few days in culture. They're beating nicely, beating all coordinated at the same time. And also when we use a calcium dye, like for example, Flipper 6, we can actually see that the microtissues light up every time that the cell contracts and releases calcium due to the excitation contraction coupling activity. Looking at the functionality, of course, not all is nice pictures and nice videos. Functionality is also quite important for these experiments. We look at the same drugs that we look, for example, in our 2D systems, like isoproterenol, nifedipine, and make sure that this also respond in the expected way as a human tissue would do, or as the 2D in vitro cells would also do. So we do treat with isoproterenol, that is a better adrenergic agonist that should increase the beating rate, and as we see in graph A, the beating rates actually increased by 50%, and also we see that the amplitude of the release of isoproterenol or of calcium also increases. We see a little bit of a shift in the baseline in the calcium traces, mostly because these aggregates or these microtissues are moving on the plate, so this produces a shift, but in general the amplitude is always changing as well. Looking at nifedipine, that is a calcium channel blocker, as we discussed before, we can see that nifedipine also decreases the amplitude of the calcium, of course, by blocking this calcium channel. So this is what we expect in a human tissue. So to describe some of the application of the Ncyte Heart in a box, until now everything is really nice. We can see that the cells are functional, the microtissues are functional, they're quite easy to put together and to assemble. They behave like we expect them to behave, but what can we use them for? So we put together a few cases or a few ways that we can show how these microtissues perform. One of them, and it's actually showing also the importance of the other cell types that are not only the cardiomyocytes, right? The cardiomyocytes play a role, but many of the diseases that we observe also have influence of other cell types that are not only cardiomyocytes, for example, cardiac fibrosis, when the fibroblasts play a very important role. We can also measure in this Ncyte Heart in a box or these microtissues elevated NT-proBNP. That shows, of course, a level of fibrosis and is correlated with heart failure and myocardial remodeling in some of the diseases like hypertrophic cardiomyopathy and all cardiomyopathies. And we can see that there's using TGF-beta1 beta as one of the TGF-beta1, sorry, as one of the triggers that will cause fibrosis. We can see that there's a dose-dependent response on the release of this proBNP. So looking at the blue graph or the 100 nanomolar, you'll see the higher release, and we compare also to ET1. That is in combination with TGF-1, of course, can be a potent vasoconstrictor that also causes cardiac remodeling. So these tissues are correctly responding to this, and cardiac fibrosis can also have other effects like release of collagen or production of scar tissue that will change also the beating of the heart, and depending on which sections of the heart this is happening, it can also bring arrhythmias and different arrhythmic events into the heart. Then we also created a model for ischemia reperfusion, and this would be something that only can be done in a 3D model. It is very hard to do, for example, in a 2D monolayer. So in the case of the ischemia reperfusion, these microtissues were incubated at different oxygen levels, starting with the normal oxygen levels of an incubator, for example, of 20%. And for 1 hour, the oxygen was decreased to 1%. The cells were also treated with different drugs, for example, sildenafil, melatonin, and vardenafil. There are drugs that will be protective of ischemia reperfusion or hypoxia in general. And when we reperfuse after 1 hour, or meaning that reperfusion, we are getting back to the normal oxygen levels, we can see that the cells recover almost to the same level that they were at the beginning. Some of the drugs having different effects that could be also sometimes detrimental in the recovery of these microtissues. And of course, there's many other applications, and some of the most important applications that we see that people are looking at now are the use of these cells or these microtissues for disease modeling. And this is why we have had a really good collaboration with the University of Leiden, where we generated cells, we generated cardiac microtissues using cells provided by the university, showing a phenotype for catecholaminergic polymorphic ventricular tachycardia. And then from that, I will pass the presentation to Richard, because he will most likely explain it much better than I can do.
Well, thanks very much, Mariana. So in my research group at the Leiden University Medical Center, we also make use of human pluripotent stem cells, and we can differentiate these into cardiomyocytes, as you can see, for example, in the video on the left of the slide, but also into various other cell types that are present in the heart. So this is including like cardiac fibroblasts, as well as endothelial cells. Now, of course, we can also generate these cells in unlimited numbers. And if the iPS cells have been derived from a patient with a particular genetic disease, then these cells can reflect the phenotype of the patient. So we can study these various cardiovascular cells either individually or in a multi-cell type culture in both 2D and 3D formats. So we can then use these cardiovascular models for a variety of applications, including for drug screening and disease modeling. And today I'll focus on our 3D cardiac microtissue model that we've been using to detect both cardiotoxicity as well as for advancing drug development. And this has been work that's been done by Tessa DeCourt and Ben Johnson in my group. So as you already saw and heard, cardiac microtissues are generated by taking the human pluripotent stem cells that have been pre-differentiated into cardiomyocytes, endothelial cells, and cardiac fibroblasts, and combining these in defined ratios to make these 3D aggregates. Now, one of the benefits of working with cardiac microtissues compared to 2D cultures is that they can be better for detecting drug responses. So for example, we performed a blind screen of 14 reference compounds. So these were drugs that covered a wide range of different mechanisms that affect contractility. And we could accurately predict the inotropic effects. So this is how the drug affects the contraction of the cardiomyocytes for more than 80% of the compounds. In particular, we found that for some compounds in which we only saw a change in the beat rate in the 2D models, when we moved to the 3D cardiac microtissue model, we could also see additional changes in other calcium parameters such as amplitude and slope time. And so this, of course, displays and gives us more information and a more accurate inotropic response. So I now want to shift focus from using the cardiac microtissues for drug safety evaluation to also how we're using them for disease modeling. And we were interested in using our cardiac microtissue model to study catecholaminergic polymorphic ventricular tachycardia, which is a bit of a mouthful, so I'll just call it CPVT for now on. Now, CPVT1 is an inherited channelopathy, which has been linked to mutations in the cardiac ryanodine receptor. Now, during situations of beta-adrenergic stress, So for example, when you're doing exercise, you get an increased load of calcium in your sarcoplasmic reticulum. Now, normally that is not a problem for us. The cardiomyocytes can cope with it. But if you have a mutation in the ryanodine receptor, this can cause the calcium to leak and result in spontaneous calcium release events, which can lead to arrhythmias. Now, if these arrhythmogenic events are not resolved, this can also lead to what's known as ventricular fibrillation and potentially sudden cardiac death. Now, unfortunately, no medication works for all people that it's prescribed to. So indeed, we see that while a particular medication will work for some, for other patients it will have no effect, or in even worse cases, it can cause an adverse drug reaction. And this is also the case for CPVT1. So between 10 to 25% of CPVT patients will still experience life-threatening arrhythmias despite pharmacological therapy. Therefore, we need to find alternative treatments for these patients. Now, this personalized approach where we aim to use patient-derived human iPSC models to identify the most suitable drug for each patient is really only possible to achieve if we can use automation and high-throughput screening technologies. So to do this, we partnered up with Ncardia with the goals of the project. Firstly, to generate a robust arrhythmia model for CPVT using our cardiac microtissues. Secondly, then we wanted to develop automated pipelines for generating these 3D models and performing the arrhythmia assay. And lastly, we wanted to perform a high throughput screen using this model to identify novel antiarrhythmic compounds. And this work has been supported by funding from Health Holland, as well as the South Holland Province, as well as the Novo Nordisk Foundation Centre for Stem Cell Medicine, otherwise known as ReNew. Now, if we want to actually be able to identify compounds that can rescue or prevent the occurrence of arrhythmias in patients, we first of all need a very reliable method for triggering these arrhythmias in these CPVT cardiomyocytes. And we've been able to identify a compound that could reliably produce arrhythmias in at least 80% of our CPVT1 cardiac microtissues, while not causing any arrhythmias in our control or wild type cardiac microtissues. So together with Ncardia, we subsequently have optimized this assay for the high throughput screen, such that at least 95% of the cardiac microtissues will develop arrhythmias. Importantly, these arrhythmic cardiac microtissues also respond and are rescued when they're treated acutely with flecainide. And now flecainide is a drug that's actually used in the clinic now to suppress arrhythmias in CPVT patients. So subsequent to this, we then developed a pipeline to automate this whole assay. So this included using robotics to generate the cardiac microtissues, then maintain them in culture for several weeks, as well as label them with the calcium dye. Then using a high throughput, high content kinetic imager, we can actually record all 384 cardiac microtissues simultaneously, as well as add compounds mid-recording to these microtissues to capture the arrhythmia rescue in real time, as you can see in this calcium trace. So as part of the assay development, we confirmed that the microtissues regenerated were reproducible. So some of the key parameters that we looked at was the size of the cardiac microtissues, and we could see that this was very consistent across 5 different plates, which were made also on different days. We also then tested the screening assay at several different time points and found that we could reliably induce arrhythmias at both day 14 and day 21 old cardiac microtissues. And importantly, also when treating them with flecainide, we could rescue the majority of these cardiac microtissues at these two time points. So overall, this sort of confirmed that our automated arrhythmia assay was reliable and indicates that we have approximately a week-long window in which we can perform compound screens. So we then moved into a pilot screen where we performed our assay on 95 pre-selected compounds, which we ran at 3 different concentrations. Then any compound that either resulted in a 100% rescue at 1 concentration or an average of at least a 44% rescue across the 3 different concentrations was considered a hit. And of the 17 compounds that we identified as hits, 8 of these have previously been used or found, tested in various CPVT studies, but there are another 9 which have not been tested before in any CPVT model. So finally, our last goal, of course, was actually to perform a high-throughput screen, and this was for over 2,000 compounds. And we performed this screen in triplicate, and we could achieve this whole screen in just 2 days using more than 6,000 cardiac microtissues. And from this screen, we've got about 100 hits to follow up. And overall, the screen had a median Z factor of 0.67, so indicating that there was good separation between our positive and negative controls, and so indicating that the assay is reliable. This is also reflected in the fact that over 50% of the hits can be broadly classified as being treatments either for cardiac or neural-based diseases. And many of the compounds that we've actually pulled out are known ion channel modulators, so of course they'll be altering the electrical activity of these cells. So the next follow-up steps for this is that we will want to perform a more detailed follow-up dose-response analysis of these hits to ideally narrow down and have a smaller number of drugs that we can then evaluate their therapeutic potential in additional CPVT patient-derived iPSCs. And we really believe that actually we're now beginning to get much closer to essentially the ultimate goal of precision medicine, which is to tailor the treatment to the individual characteristics of the patient. I'll hand back over to Mariana now.
Thank you, Richard. Very good summary of what the work was done. It looked like very little work, but it was a lot of work. So as a summary, we can see all the applications that the Ncyte Heart in a box shows, not only from the of course, Ncyte in a box and also 3D cardiac microtissues, not only from the advanced physiological relevance of this model, but also providing a cost-effective and scalable solution for more complex. Of course, the 2D can give some information, but the 3D brings the cell-to-cell interaction, the disease modeling to a different level where we can look at a more complex interpretation of the data. And this not only helps to have a more relevant model, but also, of course, accelerate the drug discovery using fully automated pipelines, reducing the cost, reducing the variability, and can help achieve more ambitious projects of precision medicine. And they can be used, as we showed, in cardiotoxicity and drug discovery assays and many all other applications that can pop into your mind. So I appreciate everyone attending this webinar, and I think that we should be ready for questions, right?
Before we go to the Q&A, I'd like to remind everyone that this webinar will be available to watch on demand at ddw-online.com. Mariana, first question is for you. So scaling up iPSC-derived cardiomyocyte production to a 10 billion cell level is impressive. What specific challenges did you face, and what quality control measures are in place to ensure batch-to-batch consistency?
That's a very good question. Honestly, the challenges are big. Of course, at Ncardia, we have done this already. We're at the point where we have a pretty standard procedure for all of our cardiomyocytes, but it is challenging, of course, to scale up, to move into bigger volumes. It brings a lot of variability. It brings a lot of risk, of course, when you're running a 3-liter bioreactor with 10 billion cells. So the risks are high, the variability is high. So the way that we tackle these kind of challenges is by being really consistent and keeping a very tight control, not only of the end product, but also throughout the whole process. So there's a lot of in-process control, looking at oxygen levels, looking at pH, metabolite production, making sure that these cells are going in the right direction by taking constant samples, analyzing trends, analyzing markers throughout the process. So of course, the quality control happens not only at the end, but during the process. So it's very important that we keep very tight control, and the process always looks the same. We also keep strict database with hundreds of runs when we keep data and analyze this data, and through machine learning, we can also understand how our process changes. So in general, we do that during the process, and at the end of the differentiation, when we are going to release our product, we also look at many things like genomic stability, contamination, identity markers, and as I mentioned during the presentation, functionality. So the cells also need to pass a strict criteria of functionality to be able to be released into the market and be used by the end users.
Brilliant, thank you. And another question for you. What role do endothelial cells and fibroblasts play in the functionality of the Ncyte Heart in a Box model?
They play a big role. Of course, not only do they create or bring that complexity that we're looking for when we are talking about microtissues, and that's why they are so important. They bring the complexity of the cell-to-cell interaction, of making the system more similar to what a human heart is or a regular heart is, but they also bring different signals into the heart. As I mentioned a little bit through when I was discussing the fibrosis, right? The fibroblasts release extracellular matrix, provide support to these cardiomyocytes, express proteins in their membrane that will be making connections with the cardiomyocytes, while the endothelial cells also produce paracrine, different substances that will be released into the media, also providing support to the cardiomyocytes. They can also produce inflammation in certain cases, and they can form vessels or vessel-like structures that will also produce or give support to this microtissue. So all of the cells are playing a different role, not only in forming the microtissue, in bringing complexity, but also in disease modeling. There might be different cells that you might want to look at when you are looking at a disease.
Okay, thanks. The next question is directed to both of you. How has the collaboration with LUMC contributed to advancements in cardiac research, and vice versa? What have been the benefits of collaborating with Ncardia?
I can tackle the first part. I think that for Ncardia, the collaboration with the Leiden University has been really valuable because, of course, we have all the experience with the cells and the automation, but the experience and the knowledge that the LUMC brought in terms of the disease modeling and how to make sure that we're keeping the right controls, that we're doing the disease modeling, and making the system the more similar to the actual disease was really important for Ncardia and brought a lot of value in that sense.
Yeah, I think prior to working with Ncardia, I mean, we as an academic group have always wanted to perform some of these assays that we've developed to establish them more in a high throughput context. And as Mariana said, the knowledge and experience that Ncardia has provided for being able to actually automate such assays has been really crucial here. And just to give an example, when we came in, as I showed, we were getting around about 80% of our cardiac microtissues showing the arrhythmia in the assay, which we were really happy with. But actually, if you want to do a large compound library screen like we've done here, 80% is not high enough. It means that almost 80 wells in your 384 well plate would be lost. You wouldn't actually get any measurements from them. So really working together with Ncardia, we're actually able to further optimize this assay, as I showed that now more than 95% of microtissues consistently we can trigger an arrhythmia in them.
Great, thank you very much, both of you. Next question is for Mariana. How easy is the implementation of this model in a new lab? How does Ncardia support that?
It is quite easy, of course. We show in this webinar the TCAN and the automated robot. We understand that that might not be the case, that everyone has access to that. So we have produced a very well described protocol, of course, for all of the people who want to buy the Ncyte Heart in a box, and we sell it as a kit of the 3 cell types in the proportions that will be used. So this application note is very well described with the protocols, with the support of exactly what needs to be used, how it can be done, with several options of assembly, and to do it by hand or automated as well. So of course the support, the online support in that case, but also of course our technical team is always available for questions, for calls, for support to really walk the teams through putting or implementing this really valuable system in their labs.
And also, what are the consequences of using patient-specific cell lines? Is optimization required?
Well, yes, optimization is required. Of course, the consequences is bringing that disease modeling that is very important. So that is, of course, a huge win when doing these kind of models, being able to use these kind of cells. And that's one of the main benefits of using iPSCs, that we really have access to all of these patient lines and disease lines, and we can use them in these models. There is optimization needed, of course, at different stages of the process. One of them is the differentiation of the cells itself. Sometimes when we work with different cells, they need a small tweak, a small optimization. Of course, at Ncardia, we have done that enough that we know which are the points that we need to tweak in different cell types, or what are the things that we can change to make the cells really reach those 98, 97% purity that we obtain in our internal cell lines. And there's also, of course, some optimization when putting together these microtissues, looking at the composition of cells. As I mentioned, we do 70% cardiomyocytes, 15% endothelial cells, and 15 fibroblasts. But the number of cells might be different if some cells are proliferating or some cells grow different. So, for example, in Ncardia, we use in our application note, we recommend microtissues of around 10,000 cells. We know that in Richard's group, they use slightly less because they're using a different cell line. So there's some optimization or some work that needs to be done, but it's not really complicated.
Okay, great. Next question is for both of you. With recent FDA news pushing further to reduce animal testing in safety and toxicity, how can this model help pharma and biotech embrace this change?
Well, I think that it brings value from several points. First of all, combining the disease modeling with a non-animal model is, of course, really valuable for this case. In general, some of the in vitro models that are available that could be primary cells might not be easy to get disease models, or cells that have certain phenotypes. So with the iPSCs, the genetic diversity and the different cell types that can be created is really, I would say, huge. It gives huge possibilities for testing different things. And it's scalable, it's reproducible. You can always get cells that look the same in big batches, as we mentioned as well, and they meet the needs for safety and toxicology studies, of course.
Yeah, perhaps I can just add, I mean, of course, these are all human-based cells, so here compared to the animal models, which are, of course, a mouse heart beats quite different to a human heart. So we can use these to really capture sort of species-specific responses to compounds, so especially, for example, related to cardiotoxicity, which might be missed in animal models. Yeah, as Mariana noted, this model is also quite scalable. I mean, it's very compatible with automated screening platforms. So it's very attractive, I think, at early stages of drug discovery as well, and all the way through to safety pharmacology. And I mean, in particular, if we're looking at repurposing drugs for diseases, I think here these human-based models are quite valuable, as of course there's already a lot known with these repurposed drugs about the pharmacokinetics, pharmacodynamics of the compound. So I hope that this can really expedite the development of these compounds to treat, or these drugs to treat other diseases, make it faster to get them into the clinic.
Brilliant, thank you. I've got some questions for you now, Richard. Based on your findings when looking at detection of cardiotoxic compounds, when would you recommend using 3D versus 2D cardiac models?
Oh, that's a good question. So we definitely see advantages with these 3D models when we're assessing if a compound has an effect, for example, on the electrical activity or the calcium handling of the cells, and therefore sort of indirectly the contraction of the cardiomyocytes. And as showed, we could pick up responses for certain compounds such as PDE3 inhibitors that we had previously missed using sort of our 2D based screens. Where sort of 3D models are still not very ideal is if we're actually wanting to look at specific effects on certain cell types in these multi-cell models. I mean, it's still quite difficult to image these cells in 3D with a very high resolution, especially if you're looking at it, doing it in terms of a screen. So here, if you're really wanting to look at an effect or examine one particular cell type, it might be better to work in these more simpler 2D formats to begin with. And then, of course, you can also combine these different cell types together in a 2D, and you also can see then the influence of these other cell types on the responses and the sensitivity of the cardiomyocytes that way.
Thank you very much for that. Next question. You have demonstrated a physiologically relevant disease assay for CPVT. Is this assay also suitable for other cardiac diseases?
Yeah, also a very good question. So we've done some extra work looking at some of our other disease lines, and indeed we found that we can also trigger arrhythmias in other arrhythmogenic diseases. So for example, long QT2, so this is where there's mutations in KCNH2, the hERG channel, and we can also again get these arrhythmias occurring in these long QT2 microtissues very reproducibly. What was also quite interesting in these preliminary studies that we've done is that we saw that the long QT2 models didn't respond to flecainide, and that would be expected. So long QT2 patients don't respond to flecainide either. Whereas we could use another compound which would rescue that arrhythmic phenotype in the long QT2 model, which didn't rescue it in the CPVT model. So we really think actually this assay can really reflect sort of the disease-specific phenotypes that would be seen with these different mutations.
Great, thank you. And last question for you just now. What are the next experimental steps to validate the 9 novel compounds that haven't previously been tested in a CPVT1 models?
So actually, of course, we've done this larger screen of 2,000 compounds where we have got another 100 hits. So we're actually going to include those 9 compounds together with those additional hits that we got out, and do a more thorough potency assay, so dose response, to narrow down to a smaller number of compounds that will then follow up with some of our other CPVT patient models that we have available, and hopefully identify a couple of compounds that will work in multiple different models that we have. We also have other methods of assaying the effects, so not only doing it in these 3D models like the microtissues, but also exploring it in either more complex 3D models such as engineered heart tissues, or also doing more lower throughput patch clamp analysis, for example, to really explore exactly what's happening in these cells in terms of the electrophysiology.
Thank you. Coming back to you now, Mariana. With the integration of iPSC-based models in high-throughput screenings, what steps are taken to minimize variability across different platforms or conditions during long-term studies?
Yeah, that's of course very important. So that's a question that is very good to ask in this context. Of course, the automation already brings down a lot of that variability, and that also helps that we have a lot of standard procedures in place. Not only for the automation is a little bit easier to have standard procedures because of course everything goes through scripts, but also for the generation of the cells, for the assembly, for the harvest, everything is a standard procedure that is validated and tested several times. We also have, of course, validation of all of our data analysis, validation of the data sets that we use, and everything is very standardized. I think that at the moment when you start working with this high throughput, having very good control of the procedures that are used, the data analysis that is used, and having the right controls in place is one of the most important things. The automation, of course, helps. In this case, when you are working with 384 microtissues, many times several places, there's going to be some variability if done by hand. As we see, the automation decreases the variability, but having good procedures and things that are very standardized also will help if the automation is not available.
Thank you. Next question. How do you ensure the optimal ratio and spatial organization of cardiomyocytes, endothelial cells, and fibroblasts during the automated formation of 3D cardiac microtissues?
Well, in general, it really helps the fact that our microtissues don't have any any matrix involved, like any of these matrix gels or anything that will restrict the movement of the cells. So when we are putting the cells together, of course, we harvest the cells individually. So it also helps that we are not letting these organoids self-assembly and let the cells going into different directions. So we are harvesting the cells, mixing them, and letting them take their place into the organoid as they form in a free will, let's say, because these organoids are really floating, so the cells align and make the connections as the organoid forms or the microtissue forms. And that's why I mentioned is, of course, you could see these microtissues beating within 3-4 days, but this cellular organization and the creation of these networks and connections in between the cells might take some more time, and the cells might still be moving around, finding their place. So that's why we recommend really using these microtissues after 14 days, when we know that that all of the cell-to-cell interactions have been formed and the microtissues are really working as they should.
Great, thank you very much. And we've got one last question from Richard. How does Ncyte Heart in a Box compare to other 3D cardiac models like EHTs?
Okay. Well, I mean, I see sort of both models as complementary to each other, but of course developed with a sort of different emphasis or for different applications. So of course, the EHTs or the engineered heart tissues are quite powerful models, especially if you're wanting to look at force generation contractility, because you get actual numbers out. But of course, they're much lower throughput. While with the cardiac microtissues, you don't get the same degree of information regarding sort of mechanical load or force, but you have the advantage of the scalability, the reproducibility of them, as well as also the opportunity to integrate them into high throughput automated workflows. So yeah, I mean, the microtissues, are much easier to produce in very large numbers, of course, and so this makes them ideal for the compound screens and profiling. And as Mariana was saying, you do still get some of the key aspects of the 3D architecture with this multicellular contribution to capture these physiologically relevant responses. So I think you can see really as the microtissues or the heart in the box as sort of a system better suited for screening and early stage decision making, while sort of the engineered heart tissues are great for other applications or further downstream when you're wanting to have more detailed contractility measurements.
Don't forget, you can listen to this webinar again on demand at ddw-online.com, and you can access Drug Discovery World content online at any time. Thank you to our speakers, and thank you for listening.